We are going to talk about the process of fertilization. Then after that, we are also going to talk about the first week of human development. We will start with the process of fertilization. If you remember, we have already talked about gametogenesis, and we have seen that gametogenesis is the process of formation of the sperm and the ovum. Spermatogenesis takes place in the testis. Oogenesis takes place in the ovary. And there are some differences between those two processes, but either way, we now have spermatozoa, and we also have the oocyte, although the development of the oocyte has been arrested at metaphase 2, but at least the oocyte has already been ovulated. So the oocyte is in the fallopian tube. Let's make an assumption that the sperm is now also within the female reproductive tract via mechanisms that we are aware about. So we want to see how fertilization takes place. Now, in this lecture, we are going to outline the mechanisms that promote the transport of the gametes, both in the male system as well as in the female system. We are also going to explain the sequence of events that occur during fertilization. We are then going to state the outcome of fertilization. And lastly, we are going to highlight some of the clinical aspects related to the science of fertilization. Let's begin with mechanism of gamete transport, and more specifically, the mechanisms of sperm transport. You realize that sperm must be transported both in the male reproductive system as well as in the female reproductive system. So let's ask ourselves, what mechanisms are there that promote the transport of sperms while they were still in the male reproductive system? Remember, sperms are formed within the seminiferous tubules of the testis, which are these ones. So there are a number of mechanisms that promote the transport of sperms from the seminiferous tubules. One of such mechanisms involves the secretions of the Sertoli cells. Remember, Sertoli cells are supporting cells of spermatogenesis. They produce some secretions. Those secretions will be flowing from the seminiferous tubules through this region, which we call the rete testis, to the epididymis. In that flow, the sperms will move with that flow. So the secretions of the seminiferous tubules, or rather secretions of the Sertoli cells, promote the transport of sperms. Also remember that within the seminiferous tubules, there are some contractile cells that we called the myoid cells. These myoid cells also contract, and their contraction will promote movement of sperms from the seminiferous tubules. The sperm has a tail which is able to beat in a particular rhythm to help the sperm to swim. Inasmuch as this motility is not very much in the male reproductive system compared to how fast it is in the female system, there is still some level of motility. So the motility of the sperm tail promotes sperm movement. And lastly, when the sperms are in the vas deferens, during the process of ejaculation, the vas deferens usually contracts. Contractions of the vas deferens during ejaculation will also promote movement of the sperms and definitely the semen from the male reproductive system to the exterior. hopefully that exterior is in the vagina. So if that exterior is into the vagina, then what happens next? The sperms are now deposited within the vagina. We know that there are many sperms which are deposited within the vagina. We talked about close to maybe 200 or 300 to 500 million sperms in a single ejaculate. How to calculate this is to just have rough estimates. We agree that a single ejaculate can be about 2 to 6 ml of fluid. Give me a second. Okay, let's carry on. So a single ejaculate has about 2 to 6 ml of fluid. The concentration of sperms per ml of ejaculate is about 50 to 100 million sperms. So if you do your arithmetics and work with an average of, let's say, 3 to 4 ml of ejaculate, then you realize that the number of sperms we're talking about could be in the numbers of 300 million sperms. At least we know that if the sperms are less than 50 million in a single ejaculate, then that is considered as oligospermia, a low sperm count. Now, out of these 300 million sperms we are thinking about, interestingly, only 1% of the sperms actually climb through the cervical canal. Now, the cervical canal is usually closed, but around the time of ovulation, the cervical canal opens in readiness for sperm entry. So during that time that the cervical canal is open, about 1% of the sperms which are deposited within the vaginal canal actually climb up the cervix. The rest of the 99% of the sperms, we can just tell them polleni. So the one that climb up, the ones which succeed to go up through the cervix, what promotes their movement? When the sperms are in the female reproductive tract, we agreed last time that they will undergo capacitation. When sperms undergo capacitation, their motility is increased. And that means that they can swim faster than they have ever swum. And so this increased motility of sperms is also a mechanism that promotes sperm movement. Also, the female reproductive tract, and this is in particular the uterus and the fallopian tube, they contract. The contractions of the female reproductive tract also promote sperm motility. You'll be told that during ovulation, although the cervix usually has some thick secretion and usually closed, during ovulation, the fluid of the cervix is more watery than mucoid. And this watery fluid is just there to facilitate the movement of the sperms. Or rather, the sperms can now swim easily within the water. You can imagine if that fluid was very thick, it would be very difficult for the sperm to swim within that fluid. But now that's watery during ovulation, the sperms can easily swim through it. All right. A question for you regarding viability of the sperms. I want you to answer it in the poll so that I'm able to monitor your performance, which I told you contribute to your assignment marks. So the question is there. You have 1 minute to answer it. Some 5 people haven't, but let me close it. So I'm projecting for you the results. The question is well answered. Okay, we've just talked about very tough competition. I saw this somewhere and I found it very interesting. So hata kama anatomy ilikuwa ngumu, just know there's a time that you actually won the race. Let's talk about ovum transport. What facilitates movements of the ovum? During ovulation, the femoral end of the fallopian tube usually sweep. That sweeping movement of the femoral end of the fallopian tube promotes the fishing of the egg into the fallopian tube. Fallopian tube is also lined by simple columnar ciliated epithelium. The simple columnar ciliated epithelium of the fallopian tube promotes movement of the oocyte from the femoral end of the fallopian tube towards the uterine end of the fallopian tube. There are secretions within the fallopian tube with the hope that these secretions are flowing from the femoral end to the uterine end. These secretions can also facilitate movement of the oocyte. And lastly, the fallopian tube usually contracts. The contractions of the fallopian tube also facilitate movement. Now, maybe in this case, it might not be the egg per se, but now the conceptus in the assumption that fertilization has occurred. This is particularly important when the conceptus is traversing what we call the isthmus of the fallopian tube, which is a relatively narrow zone of the fallopian tube. The contractions of the fallopian tube facilitate the transport of the conceptus through this narrow segment. So these are the mechanisms that promote gamete transport in female. Now we want to talk about the process of fertilization in the assumption that the sperms have found themselves in the female reproductive tract and maybe not the rectum or in the esophagus. Fertilization is a fusion of the male and the female gamete to form the zygote. This process takes place within the ampulla of the fallopian tube. This is the normal site of fertilization. Perhaps you're wondering, what are we calling the ampulla of the fallopian tube? Let's take this to be the female tract, with this being the uterus and this being the fallopian tube. The other name for the fallopian tube is the oviduct, a relatively older term though. We can call it uterine tube or fallopian tube. So let's see, this part of the fallopian tube is what we call the fimbria of the fallopian tube. The fimbria of the fallopian tube is the one that sweeps during ovulation to promote the fishing of the oocyte that has been ovulated. Remember, the ovary is somewhere here, and actually there is no direct tubular connection between the ovary and the fallopian tube. So it's a lot of chance, but there's some guides. The sweeping movement of the fimbria promotes the chances of the oocyte entering the fallopian tube. That region of the fallopian tube is known as the infundibulum of the fallopian tube. The infundibulum of the fallopian tube is funnel-shaped, as we can see here. The term infundibulum means funnel, actually. This is what we call the ampulla of the fallopian tube. It's a relatively longer segment of the fallopian tube, and that is the site of fertilization. This narrow segment of the fallopian tube is what I've termed the isthmus. And this is what I was telling you, that for the egg to move from here, it will require some contractions of the fallopian tube because this is a narrow zone. Now, there's still a part of the fallopian tube that is traversing the uterine wall, perhaps from there to there. We can't call it the uterus. It's a lumen, the lumen of the fallopian tube that traverses the uterine wall. We call it the intramural segment of the fallopian tube. So these 5 are the parts of the fallopian tube. How about parts of the uterus? This upper part of the uterus is what we call the fundus of the uterus. This is the body of the uterus, and this is the cervix. This region here is what we call the endometrial cavity. This region here is what we call the cervical canal. The cervical canal has 2 openings. The upper opening is called the internal os. The lower opening is called the external os. The junction between the body and the cervix is called the isthmus of the uterus. Same spelling as this one, except this is isthmus of the fallopian tube. This is isthmus of the uterus. The junction between the fallopian tube and the uterus itself, that corner, is what we call the conus of the uterus. Conus means horn, in plural conio. All right, so those are the parts of the uterus as well as parts of the fallopian tube. Fertilization occurs here within the fallopian tube, the ampulla of the fallopian tube. Before we talk about that process of fertilization, I want to remind you about the Graafian follicle just before it is ovulated. So this is how mature follicle looks like. Now, that region labeled A represents the secondary oocyte. It is the egg that has completed the first meiotic division, but while it started the second meiotic division, it got arrested at metaphase 2, secondary oocyte. That arrest takes place a few hours before ovulation. The covering of the oocyte, whether primary or secondary, is what we call the zona pellucida. It's a glycoprotein covering. This other layer here is the basement membrane of the follicular cells. These are the follicular cells. As you can see, it's a stratified epithelium. That's the basement membrane of this stratified epithelium. Now, these cells, and these cells, I've said, are the follicular cells. but the follicular cells exist in 3 different populations. The population labeled D is what we called the coronary.
Coronary radiata.
Okay. I think that was a break from Marion. So we're saying this, that these cells here, the follicular cells, the population of follicular cells that are around the zona pellucida is what we call the corona radiata. The population of follicular cells which suspend the oocyte, these ones are the ones we call the cumulus euphorus. This region is the follicular antrum. This cavity is a follicular antrum. The population of cells external to the follicular antrum, these ones, are the ones we call the granulosa cells. So granulosa cells, cumulus euphorus, and the corona radiata are part of the follicular cells. Remember that this region here, external to the basement membrane, is what we call the thicker folliculi. The thicker folliculi has 2 zones. There's this inner zone which contains cells. We call it the thicker internal. And then there's this outer zone that is relatively fibrous. We call it thicker external. So these are the parts of the Graafian follicle before ovulation. When ovulation takes place, this is what is ovulated. That means that cumulus euphorus will cease to exist. We ovulate the oocyte, the zona, and the corona en masse. The remainder of the follicle constitute the corpus luteum. The cells of the corpus luteum will then be called the granulosa lutein and the thicker lutein. Okay, let's focus on what has been ovulated. So this is what is ovulated. We have the corona radiata, secondary oocyte, and the zona pellucida. The first thing that must happen during the process of fertilization is that the moment the sperms are deposited in the female tract and they are lucky enough to climb up the cervical canal, they'll undergo capacitation. We describe capacitation as the removal of the glycoprotein covering from the head of the sperm. This process of capacitation has twofold benefits. One is that they make the sperms more motile, which means that the sperms can now swim faster towards the oocyte. But importantly also, the identity molecules of the sperms are unmasked. And that means that the sperms are now able to identify, at least from a molecular level, they're able to identify the oocyte. So capacitation of sperms take place. The sperms will then swim up to where the oocyte is. And once they reach there, the sperms will then penetrate through the corona radiata. These are the cells which cover the cells which cover the zona pellucida. These are corona radiata. They are follicular cells. So the sperms will swim through. It's still a hard task. I forgot to tell you that another hard task was swimming through the fallopian tube. Remember, we mentioned that the ciliary activity of the fallopian tube is from the femoral end to the uterine end. That means that the sperms are swimming against the resistance of the ciliary activity. Kazi. All right, so once the sperms have penetrated the cells of the corona radiata, what next? They will make contact with the zona pellucida, like that one. The moment the sperms establish contact with the zona pellucida, the sperm will release some enzymes from the acrosome. The process of releasing enzymes from the acrosome is known as acrosome reaction. What is the importance of acrosome reaction? Let's see here. So this is the sperm that has made contact. This is the sperm that is releasing enzymes from the acrosome. The importance of the acrosome reaction is to digest the zona pellucida. Remember, zona pellucida is a barrier. The sperm need to penetrate this barrier so that it can enter the oocyte. For it to penetrate through this barrier, it must break through. So that process of breaking through is aided by the enzymes from the acrosome. Now, the zona pellucida has multiple layers of barrier. There's a gel, and there's another coat there, which we call the fertilization envelope. There's some proteins within it as well, and receptors, as we can see. But that's not the intention of this talk. The point here is that the acrosome releases enzymes that will help the sperm to penetrate the zona pellucida. And so, sure enough, it penetrates the zona pellucida. After the sperm has penetrated the zona pellucida, the cell membrane of the sperm will then fuse with the cell membrane of the oocyte. After the cell membrane of the sperm has fused with the cell membrane of the oocyte, as we can see in that step, it means what? That the nucleus of the sperm will then be extruded into the cytoplasm of the oocytes. Once that happens, a number of things will take place. Importantly, something called zona reaction will occur. Now, what is zona reaction? Zona reaction is the biochemical hardening of the zona pellucida. The biochemical hardening of the zona pellucida is called zona reaction. Why does it harden? It hardens so that it cannot be penetrated by another sperm. So when it hardens, the sperms which were penetrating through it will be entrapped within that, so they'll not go anywhere. Hiyo ni tofauti na kunyongwa. The other thing that will happen is that the oocyte usually has some granules just beneath the cell membrane. We call them cortical granules. These cortical granules will also be released in a process we are calling cortical reaction. That cortical reaction also establishes a very thick barrier between the zona pellucida and the cell membrane of the oocytes. This very thick barrier also prevents polyspermy. Now, what is polyspermy? Polyspermy is fertilization by more than one sperm. So the aim of the zona reaction and cortical reaction is to prevent fertilization by more than one sperm. The moment the nucleus of a single sperm has entered, these changes occur within minutes, so that it forms a barrier that another sperm cannot penetrate again. And that is also what is depicted in these images. The moment the nucleus of the sperm enters, cortical reaction takes place, zonal reaction takes place, so that now no other sperm can enter. Perhaps I ask you a question at this point. And this is the question. What would be the outcome of two sperms fertilizing a single oocyte? I want you to answer it in the poll. Okay, the poll is now with you. Just answer that. You have 1 minute for it. All right, okay. All right, everyone has. So this one is not well answered. Before I tell you the correct answer, perhaps let's think through. So maybe I get 2 or 3 people who volunteer what they wrote and why they chose it. Any 2 or 3 people. You can, let me allow you to unmute yourself. Then you can unmute yourself and respond. You should be able to unmute yourself now. Anyone?
I picked on none of the above.
Okay.
This is because 2 sperms will be accepted into the egg, but only one will be fertilized. Only one.
Okay. Now we are saying that 2 of them actually fertilize the oocyte. Let's take that scenario that 2 of them have actually fertilized the oocyte. But I get your argument. Another person?
I think it is trisomy syndrome.
Okay, why?
Because I think here you look at the number of chromosomes, because each sperm will contribute each one chromosome and the oocyte 1 chromosome. So I think it is, you look at the number of chromosomes.
Okay, I get your point. How many chromosomes does a single sperm have?
23.
23, good.
23.
So 2 sperms, each has 23 chromosomes. Then the oocyte, which also has 23 chromosomes. How many chromosomes will the zygote have? Anyone?
46.
Yes, Abu ulisoma wapi bwana?
69.
69.
69.
69.
69.
69.
When we talk of trisomy, we mean how many chromosomes? A trisomy
47.
47. Monosomy syndrome has 45. So at least we are sure it's not C and it's not D, because the zygote here has 69 chromosomes. we will learn how twinning come about and how triplet pregnancy come about. But a zygote which has 69 chromosomes cannot be a human being. So it can't be twin, it can't be triplet. The correct answer is none of the above. We will see what it will become, but at least not these 4 options. So that brings me to the role of the zona pellucida. Why do we have a zona pellucida? Zona pellucida has a number of functions. One is to prevent polyspermy, which is fertilization by more than one sperm. And we have seen that if more than one sperm enters, then you will affect the ploidy. Remember, a single oocyte is haploid. A single sperm is haploid. The zygote should be diploid. If 2 sperms enter on a single oocyte, then that will be triploid. When you have polyspermy, you have polyploidy syndromes. Could be triploidy, could be quadriploidy, depending on the number of sperms which have entered. And we are going to see the outcome of polyspermy. The other role of the zona pellucida is to ensure that the sperm that penetrates the oocyte is of the same species as that of the oocyte. You've seen some proteins on the zona pellucida. Those proteins are unique for each species. So only sperms of that species will penetrate that zona pellucida, hence that oocyte. It ensures that if you're a human being, now all the ladies have said no, if you decide to go to a cow, you shouldn't have an offspring. It ensures species specificity. There are some situations where that has been breached, of course, where a good example is the scenario of a mule, which is the offspring of a horse and a donkey. But you realize these 2 animals are very close together. So perhaps the identity molecules are actually similar in that sense. So the zona pellucida is not able to distinguish the 2 species. All in all, the mule, the resultant animal, is usually infertile. Nature has a way of ensuring that such a species cannot give rise to its own kind. We are also saying that the oocyte must have stayed very long before it is fertilized. If you remember that story well, the first meiotic division started before this girl was even born. Then this egg is going to be ovulated, let's say when the woman is 45 years. This egg has actually seen all the presidents of Kenya. It's very old, the egg. And so if the egg stays that long, there are higher chances that some chemicals can enter it and distort its genetic makeup. The zona pellucida forms a barrier to such chemicals so that the genetic makeup of the oocyte is not destroyed or altered. There are no mutations. That doesn't mean that we achieve 100%. And so it explains to you why also genetic disorders are more common with increasing maternal age. If 100 women in their 20s gave birth and 100 women in their 40s gave birth to children, it is possible to find a higher prevalence of genetic disorders in the children who are born to these mothers who are in their 40s compared to the children who are born to the mothers in their 20s. I will not say the moral of the story is Maliza Haraka, but you get the point. Now, it is important that you realize that when zona, okay, that should be zona reaction, not acrosome reaction. When acrosome reaction occurs, it occurs when only the head of the sperm has entered. That means that only the head of the sperm enters. The rest of the sperm is cut off at the neck. Na si uchungu. Now you realize where this thing of naingishwa kichwa peke yake came from. Even the sperm only enters the head. Fine. After the head of the sperm has entered, then and zona reaction occurred, it is at that time that now the oocyte can complete its second meiotic division. Remember, the oocyte was arrested at metaphase 2. So now the oocyte completes its second meiotic division. And once it has completed its second meiotic division, it forms what we call the female pronucleus. On the other hand, the nucleus of the sperm is called the male pronucleus. So you have two pronuclei in a single cell. The male pronucleus then fuses with the female pronucleus to form the zygote. That stage of development when you have two pronuclei in a single cell is referred to as the otid stage. The otid stage is when we have the female pronucleus and the male pronucleus. They have not yet fused, but they are present in a single cell. We call that the otid stage. So the two pronuclei fuse to form the zygote from the otid stage. That is the process of fertilization. Maybe, you know, it was a very difficult journey for the sperms. Maybe some of the sperms said, ngojeni hapa narudi. Whichever works. What is the outcome of fertilization? What are the results of fertilization? The zygote is a result of fertilization. Unless the sperm enters the oocyte, we can never form the zygote. The other result of fertilization is this. We had mentioned this earlier, that the oocyte can only complete its second meiotic division if it is fertilized. And what happens when it is fertilized? It will complete the second meiotic division, and that means you'll form the mature oocyte, and the extra genetic material that will be shed off is what we call the second polar body. So second polar body can only be formed when second meiotic division is completed. Also, the diploid number of chromosomes, the 46 number of chromosomes, is restored at fertilization. The oocyte is having 23 chromosomes, haploid. The sperm also has 23 chromosomes, haploid. When the two shake hands, the zygote has 46 chromosomes, diploid. When a woman is ovulating, she cannot tell, ati now this one, the way kana niuma hivi, aka katakuwa kaboy. It's not possible to tell the gender of the oocyte before ovulation. But after before fertilization, it's not possible to tell the gender. You don't know whether this egg will be male or female. But after fertilization, we are able to tell the gender. Now, this gender is not necessarily the anatomic gender, but the genotypic sex, where we say 46XX or 46XY. That we can tell after fertilization. We couldn't tell it before fertilization. Determination of genetic sex, you can call it embryonic sex. Now, you took half of the genes of your father and half of the genes of your mother. Then you made a concoction of yourself in the process of fertilization. That means that you are actually different genetically from any of your parents, so to speak, because you only took half from your father, not everything. You only took half from your mother, not everything. And I am assuming that your father and mother are not relatives. Forgive my assumption. Now, if they're not relatives, they're genetically different, then you take half of your father, half of your mother, then you make a concoction of yourself. You are definitely different from any of them. And that's what we mean by genetic variation. We talked about cells having undergone cell division and losing the capacity for such process again, cells entering G0 phase. The oocyte is a good example of such a cell that loses the capacity for cell division ever again. However, if the oocyte is fertilized, it becomes metabolically activated, and the machinery responsible for cell division is actually activated, so that that capacity for cell division is restored at fertilization. The type of cell division that the zygote will then undergo is mitotic cell division, not meiosis, but mitosis. The mitotic cell division that the zygote undergoes is hereby termed cleavage. Cleavage is actually a series of mitoses that characterize early developmental stages, not limited to the zygote only. That series of mitotic division that characterizes early developmental stages is what we call cleavage process. So the process of cleavage is activated at fertilization. All right. As we finish on the process of fertilization, let's talk about some 3 clinical aspects of fertilization. We'll talk about contraception, we'll talk about aneuploidy, and we'll talk about what we call molar pregnancy. Let's start with contraception. Contraception is deliberate use of artificial methods or other techniques to prevent pregnancy as a consequence of sexual intercourse. There are multiple methods of contraception, but as with regard to fertilization, the key is to prevent the process of fertilization from taking place. So in as much as I'm going to list for you multiple methods of contraception, realize that some of them may not necessarily be blocking the shaking of hands of sperm and ovum, but most of them do that. So things like coitus interruptus. This is the story of usimwage ndani. Coitus interruptus is the withdrawal of the penis just before ejaculation. The aim here is to ensure that the sperm and the ovum will never meet, in as much as the other 2 organs met. So it applies on preventing conception. Barrier methods, things like the condoms, female condom, male condom, they just prevent fertilization. Well, you are aware that the barrier methods also prevent against sexually transmitted diseases, but we are talking about fertilization here. Sterilization methods are methods which are deemed as permanent surgical interventions to prevent pregnancy. They are considered permanent contraceptive methods. For the male, it involves cutting of the vas deferens, and that is what we call vasectomy. For the female, it involves tying and cutting of the fallopian tubes. Remember, there are two tubes, so you cut both of them. That's what we call bilateral tubal ligation. They are considered permanent methods. So a woman opts for this when she has no intention whatsoever of reverting fertility. You are advised that you only do this if you know that you already have the desired children number. Or maybe if the children are 10 already. Fertility awareness methods are largely based on a woman knowing her cycle so that she avoids unprotected sex during her fertility window. The fertility window revolves around the timing of ovulation, still preventing the process of fertilization. Emergency or postcoital contraception involves use of progestin-only drugs. This one, a woman takes like within 24 to 48 hours after unprotected sex. If this happened during her fertility window and she fears that she might be pregnant and she didn't plan for it, she can take these pills, which we call e-pills. Now, the concept is that these drugs have high levels of progestin. When the progesterone levels are high in the circulation, two things would happen to this woman. If this woman has not yet ovulated and had sex, maybe she was going to ovulate tomorrow, she has sex today. If she takes that drug today, this drug will inhibit the surge of the luteinizing hormone from the pituitary gland. And if you inhibit the surge of if you inhibit the surge of luteinizing hormone from the pituitary gland, you're going to prevent ovulation from taking place. So that's one mechanism. By inhibiting luteinizing hormone surge, hence there'll be no ovulation that particular cycle, or at least delayed. But there's another mechanism, and this is especially helpful now if the ovulation has already taken place, in as much as it's still helpful even before. Perhaps the stronger mechanism. Progestin makes the cervical mucus be very thick. Remember I told you during ovulation, the cervical canal has thin fluid, watery. If a woman takes progestin, the cervical mucus becomes very thick. And so the sperms cannot swim. Now you can imagine swimming in water vis-a-vis swimming in porridge. The sperms will not go far. So that's how the e-pill works. They are progestin-only drugs, but they're also hormonal drugs. Well, the e-pill is a hormonal drug, but there are still other hormonal drugs which can be implants or combined oral contraceptives. They have both estrogen and progesterone. They work through different mechanisms. Perhaps most of them actually still preventing fertilization. Then there are others like intrauterine contraceptive devices. These ones may not necessarily prevent fertilization, but they prevent implantation. If a woman has given birth and she's breastfeeding, that can also be a form of contraception on itself. The reason, the point here is that when a woman has given birth and is breastfeeding, she has high levels of prolactin hormone. Prolactin hormone usually inhibits ovulation through maybe a longer mechanism, but eventually a woman who has high levels of prolactin might not necessarily ovulate. Now that's not 100%, and especially it's usually just a narrow period, maybe the first few months after delivery, maybe less than 6 months or maximum 6 months. The conditions here is that the woman must be breastfeeding to ensure that you have high levels of prolactin. Okay, that's the first clinical entity, contraception. The second clinical entity I want to discuss is aneuploidy. Aneuploidy is numerical chromosomal disorders. How do they come about? They result from non-disjunction of chromosomes or chromatids during meiosis. The chromosomes fail to separate, or the chromatids fail to separate during meiosis. So this is called non-disjunction. It means that the gametes, peradventure, one gamete may have 22 chromosomes instead of 23. Another gamete might have 24 chromosomes instead of 23. If such gametes then participate in fertilization, and in this case, they meet with a gamete that has normal chromosomal number, then you can actually get some abnormalities. If a gamete with 22 chromosomes fuses with a gamete with 23 chromosomes, the resultant zygote will have 45 chromosomes, and that's a monosomy syndrome. If a gamete with 24 chromosomes fuses with a gamete with 23 chromosomes, the resultant zygote will have 47 chromosomes. That's a trisomy syndrome. I don't know whether this is good news or bad news, but most of the aneuploidy syndromes actually terminate as miscarriages. I'll leave it to you to know whether that's a good news or bad news. But most of them terminate as miscarriages. There are few of the aneuploidy syndromes that do not terminate as miscarriage. Some of these actually, the children are born, but again, they die within the first few weeks or months. But there are those that actually are compatible with life. You have another question here. So this is your question. Sorry? Right, that's a question. I'll prefer that you answer it in the poll. Let me launch it for you. Okay, it's there. So you have 1 minute for it. Okay, I think I'll close it there. 2 people didn't respond though. So this one is clearly not well done because more than half of you have failed it. I'm not so sure why. I gave you an assignment on cell cycle and cell division. I was hoping it will be easy for you because you did the assignment. So you'll go check why, but the correct response is anaphase 1. Now, I'm presenting to you some normal human karyotypes. These ones, this individual has 46 chromosomes in total, with the 23rd pair of chromosomes being XX. So this is normal female. This other individual has 46 chromosomes with the 23rd pair being 1X and 1Y. So that is male, normal. That you know. This other one shows you trisomy 21. So this is an aneuploidy syndrome. Trisomy 21 is what we call Down syndrome. This is showing you the 47XXY. And I hope you remember why we don't call it trisomy 23. 47XXY is the Klinefelter syndrome. This is showing you 45X0, or you can call it 45X. Again, you know why we're not calling it monosomy 23. This is Turner's. On your right, we have 47XXX, which we call triple X syndrome. Here on your left, you have trisomy 18, now known as Edwards syndrome, and trisomy 13, Patau syndrome. But then Patau may not actually be so compatible with life, but the children be born maybe a few months, then they pass on. But yeah, they die perinatal life. We can have 47XYY. These ones actually live to term. They can actually live into adulthood. This is what we call super male, or others called Jacob syndrome. On your right, we have trisomy 8. They don't survive so much, perhaps die in the first few months, maybe the first month or the second month after birth. Now, I want to ask you a question with regard to 47XYY. And I want you to think through this before you answer. I'll actually give you 2 minutes. So XYY, for you to have XYY, it means there was non-disjunction. The question is, at what stage was that non-disjunction? Those are your choices. So again, answer it in the poll. You have it now. Right, there are few people who are yet to answer. Please do so. But I'm closing it now. I think 5 people haven't answered. Okay, now 4, now 3, now 2. Oh. I don't know whether people are struggling with cell division and cell cycle, but I just told you that maybe you didn't understand even the concept of the first one. When we talked about non-disjunction and we chose anaphase 1, the reason is because chromosomes and chromatids separate during anaphase. So by all means, your choices should only revolve around anaphase, nothing else. Now the debate here is it anaphase 1 or anaphase 2? If you chose any other choice apart from something anaphase, you are really, really off. I want one volunteer to tell me why anaphase 1 or why anaphase 2. The others are wrong, just like east is from west. The answer lies here. Is it anaphase 1 or anaphase 2? Any volunteer, you can unmute yourself and talk. Okay, we'll proceed, no volunteer. When you have XYY, it cannot be anaphase 1 because you have X and Y chromosome separating during anaphase 1. But you have the chromatids of the Y chromosome separating in anaphase 2. So if the two chromatids go on one side, that is when you can have XYY. You can only get XYY on the second meiotic division, not during the first meiotic division. Because in the first meiotic division for the sperm, you are separating the X and the Y sperm. So one secondary spermatocyte goes with X and another secondary spermatocyte goes with Y. Then now during anaphase, during second meiotic division at anaphase 2, ideally the chromatids of the Y chromosome are the one that should separate, so that one chromatid here, one chromatid here. So if that doesn't happen, then you have two chromatids in one cell. If it participates in fertilization, then you get XYY. You have to think through that one. Let's talk about the third clinical entity of fertilization. And this is molar pregnancy. What is molar pregnancy? Molar pregnancy is also known as hydatidiform mole. It is a pregnancy that is characterized by vesicular proliferation of placental tissues. The vesicular proliferation of placental tissues may be with some embryonic or fetal parts, or maybe the embryonic or the fetal parts are not there. When there are some embryonic or fetal parts, we call it partial hydatidiform mole. When the fetal parts or the embryonic parts are absent, then we call it complete hydatidiform mole. How does it come about? It comes about when the set of the paternal genome is more than the set of the maternal genome. There's some terms here that need to be clear for you. A set, what does a set mean? A set refers to 23. One set has 23 chromosomes. So a zygote has two sets of chromosomes. One maternal origin, another paternal origin. A sperm has one set of chromosomes, all of them paternal origin. Oocyte has one set of chromosomes, all of them maternal origin. So if the zygote has more paternal sets than maternal sets, then you get molar pregnancy. Now I want you to start asking yourself, therefore, how can this be possible? There are three ways in which it can be possible. One of them is polyspermy, the question I asked you earlier. If an oocyte is fertilized by more than one sperm, let's say an oocyte is fertilized by 2 sperms, then it means that the zygote will have a genome with excess paternal chromosomes than maternal chromosomes. If there are 2 sperms specifically, then the oocyte will be triploid. But this triploidy would be that 2 sets are paternal, 1 set is maternal. So that the genotype of the zygote would be 69XXX. That would apply if the sperms that fertilize that oocyte were both X sperms, or 69XYY if the sperms that fertilize the oocyte were both Y sperms. You can still have 69XXY if the sperms that fertilize that oocyte are both X and Y. Remember, I have given you a scenario where the sperms that entered were 2. If there are more than 2, then of course you'll not have a 69. This type of molar pregnancy will give you partial molar pregnancy. There'll be some fetal parts because there is evidence of maternal genome. As long as the maternal genome is present, it will be a partial molar pregnancy. Second cause would be if, let's say, a single sperm enters the oocyte, yes, but then the sperm decides to replicate its genome before fusion. A single sperm entered, but the genome of the sperm is replicated before the male pronucleus fuses the female pronucleus. You are still going to have molar pregnancy. In this case, the embryo can either be 69XXX or 69XYY. The concept here is that if the Y sperm is the one that entered, then it will be 69XYY. If the X sperm is the one that entered, then it will be 69XXX. It was a single sperm replicating its genome. The third possibility, so this second possibility will also give you partial molar pregnancy. But this third one will give you complete molar pregnancy. How does complete molar pregnancy come about? In complete molar pregnancy, either it's 1 or 2 sperms which have entered, but the oocyte is genetically empty. I'm using this term to mean that the genome of the oocyte is not there. Either it got lost or it was not there completely. Either way, the oocyte is genetically empty. So what will that mean? In most cases, if it's a single sperm that entered the oocyte and the oocyte is genetically empty, or the genome of the oocyte is rejected, so to speak, in most cases, that single sperm will replicate its genome so that it can still get the 46. But if it were 2 sperms that entered, there'll be no need to replicate the genome. So either way, you realize that the genome of the zygote would be 46. But of importance is that all of them are paternal in origin. This type of molar pregnancy will give us complete molar pregnancy. So once again, when we talk of molar pregnancy, what happens that type of pregnancy will be characterized by abnormal proliferation of the placental tissues. You may have fetal tissues or you may not have fetal tissues. If you have fetal tissues, then it means that the maternal genome was present. We call that partial molar pregnancy. If there are no fetal tissues, it then most likely means that the maternal genome was absent for whatever reason. And that's what we call complete molar pregnancy. Great. So I'm done with the science of fertilization. We've also talked about the clinical aspects of the science of fertilization. I'll give you a break. Let's see. So it's now 5:15, taken long. Maybe 10 minutes break. Take 10 minutes break, then we come back. We talk about first week of development